So, in the mouth, carbohydrates start to get broken down. Only carbs. In the stomach, only proteins. But in the small intestine, all of them, including fat. So fat hasn't been broken down yet until it gets to the small intestine. It gets broken down into, again, fatty acids and glycerol with the help of an enzyme called lipase. Here's the word for what bile does to those fats. Breaking them up into small little droplets is called emulsification. So anyway, that's that. But you notice there's no green or red stuff here except for the term lipase appeared in that last slide in the red column, which means know that that enzyme secreted by the pancreas into the small intestine. So here is a good cutaway picture of your small intestine with a little bit of an enlargement and then another enlargement over to here. All kinds of how structures relate to function stuff here. This is a classic example of surface area increasing the efficiency of some kind of process that involves surface. In this case, it's absorption. Okay. However, in red here, notice that they tell you that the contraction of circular and longitudinal. I've seen them ask you this in a fill-in-the-blank kind of style. The contraction of blank and blank muscle of the small intestine. Circular and longitudinal. What's that mean? Well, here's the two muscle layers, right? You have these muscles that are arranged, that would be these, circularly. So if you look at my hand now in the picture, that would be like my fingers here. They're arranged circularly around this tube. What happens when they contract? They squeeze, and whatever is in there is got to go somewhere else. What do the longitudinal ones do? When they squeeze, they make this a little shorter. And when they relax, they make it a little longer. A combination of the contraction of those two is what moves food through your digestive system. You can feel this sometimes in waves of contractions called peristalsis. So it's kind of like, to make an analogy, if you took a tube of toothpaste, okay? The toothpaste is your lunch. The toothpaste tube is your intestines here. If you went to the bottom and you squeezed it, right? Whatever toothpaste that was in that bottom part now gets moved a little further up, right, towards the end, or a little further down towards the end of this, which is the anus, right, in this case. If you then move your fingers up an inch or so and squeeze again, you further continue the movement of that toothpaste up and out of the toothpaste tube. So by starting at the bottom and then a little higher, a little higher, a little higher, a little higher, you could, in a series of contractions, move all that toothpaste out of the tube. That's what happens here, right? First, this part of the tube contracts, squeezes stuff this way, then this part contracts, then this part contracts. This is what is known as, again, peristalsis, described as a wave of contractions. First here, then there, then there. Okay, but now we look in the middle part. Oh, sorry. Again, specific thing here. This doesn't just move your lunch along. It also squishes it around and mixes it with enzymes and makes it more likely for each molecule to bump into an enzyme. Okay, and so here now, look what they did here. This is not like them to be this specific. The tissue layers, they want you to identify, possibly again in a diagram like this one, right, are longitudinal and circular muscles. And again, to identify the two, you'll see little lines. If those little lines are lengthwise, those are longitudinal. If they're like going around in a circle in this layer, they would be circular. But then you have a layer that's called mucosa. That would be this kind of light tan layer here. And then you have the outermost layer, just like it is with your skin, called epithelium, right? And they specifically threaten to ask you those. Okay. Now, they have something on the next page, which is also reflected in this green one here, that there are 3 levels of folding, actually 4 if you want to count it, that make this small intestine really good at digesting all your lunch and absorbing it. Those 3 levels of folding are like this. If this is your mouth, right? And like we did before, if this was your whole digestive tract from mouth to anus, then your body would only have the opportunity to move your lunch actually into your body to get to your cells in this much surface area if it was one straight tube, right? But since what actually happens is this tube goes down for a while, but then it goes back and forth and back and forth and back and forth, right? Now it's got about 21 feet worth of tube, as opposed to what would it be if it just went mouth to anus? It would be, 3 feet, 4 feet, depends on how tall you are, right? So that's the first level of folding that increases the surface for absorption in this case. What's the second level? If you look inside this tube, cut it open like they did here and look inside, you'll see its walls are kind of folded. So again, these folds make all these layers where more of your lunch can again bump into this absorptive surface. But then if you look at an even smaller level, right, which is what they magnify here, one little clump of one of these bigger folds, it shows that the actual epithelial lining is folded into finger-like, this much like shag carpet or your fingers, right, that are called villi, which is really hard to read here because the I's and the L's look all the same, that are called villi. So you can see that this villus, that would be singular, villi plural, this villus is well constructed to do what it does. Absorb stuff, more surface area than if this lining didn't project out, finger-like style. But also, where's your lunch headed? It's headed into your bloodstream so that it can take it to all your cells from the top of your head to the tip of your toes. Where's the best place to put these blood vessels? Right across from this single layer of cells called epithelium. Here now we get to the last level of folding. If you look at single epithelial cells like this one, you see that the side of them that faces the opening, called the lumen, that's where your lunch is bumping into it, itself has little tiny villi. They could have shown this a little more magnification. It has little tiny villi that make it look like it's kind of like a brush, which is why somebody called it a brush border. We could have done with that name. But what's a good name for little small villi? Microvilli. So it incredibly increases the surface area of your intestines compared to a straight tube. The curving of the tube, the folding of the inner lining of it, the folding of that lining into villi, the folding of the edge, the membrane, the actual phospholipid bilayer membrane of each villi and the microvilli, that makes the actual surface area of your small intestine something like half a tennis court or something like that. It's one of those ridiculous kind of comparisons. So, this is red, okay? This is what we just got finished talking about. I couldn't fit it into the last page, but the villi increase the surface area of that outer layer, that inner outer epithelial layer of cells, right? And this is a cross switch. Absorption is carried out, so more surface area, more absorption, right? What are they going to be absorbing? Little pieces that are small enough to be absorbed, right? Not just the organic monomers like monosaccharides and fatty acids and things, but also things like calcium ions, minerals, and also vitamins, okay? And so again, when they put this in the syllabus, they could turn this into a word-for-word fill in the blank thing, blank absorb da-da-da-da-da, fill in the blank is villi, Or villi absorb blank monomers, mineral ions, vitamins. They could leave any one or all of those blanks. Okay. Different methods of membrane transport are required to absorb different nutrients. This goes back to more review. What are they talking about? Passive transport. Like what? Like simple diffusion. What did that mean again? You're going through the phospholipid part of a cell because you are maybe a fatty acid and you're nonpolar. That would be an example of simple diffusion. What would be facilitated diffusion? If you have some kind of protein in the membrane that helps you diffuse through, but you're still going from where there's a greater concentration more to a lesser concentration across, net movement. We're reviewing all that stuff, right? Or it might be actively transported. Another kind of protein actually changes its shape and throws things either in or out, in this case in, out of your intestine into where your blood vessels are and where all your cells are.